Hi, my name is Jafar Anjad. I am doing my Master of Science in Medical Microbiology at Bushehr Medical University. In this video, I'm going to share with you about the evaluation of adherence of vibrio cholera Non-O1, Non-O139 to HT-29 cell lines. Bear with me. Successful establishment of infection by bacterial pathogens requires two adhesions host cells. Bacteria come together when they threaten, when a strong immune system responds to bacteria by producing cytokines, chemokines, and other reagents that are able to destroy the bacteria. These bacteria have a choice. They can either stay as an individual cells and die, or they can change their form and instead of being a planktonic bacteria, form biofilm. Acute bacterial infection and chronic bacterial infection are very different. Acute bacterial infection are caused by planktonic bacteria that are bacteria that is swimming freely. Chronic bacterial infection, for the most part, are caused by bacterial biofilm. Bacterial biofilm is an essence of multicellular organism, so that each individual bacterium within the biofilm is an analogous to each individual cells in your body, so that actually functioning as a single organisms. Vibrio cholera have two life forms in human intestinal. In one form, bacteria exist as single organisms, whereas in the other form, bacteria organize into sessile aggregation. The latter form is commonly referred to as a biofilm growth phenotype. Bacteria for this form needs to add to human intestinal and expression the virulence genes and expanding the evading of the human immunity. In this study, we evaluate of the adherence of vibrio cholera to human intestinal cells, HT-29 cell lines. Successful cell cultures depends upon many factors, including the quality of the cells and reagent you use, your sterile technique, the amount of experiment, the right laboratory equipment. The basic equipment of the cell cultures laboratory, including the cell cultures hood and incubators, a refrigerator, freezers, a hemocytometers, and a microscope. You will also find the cell cultures flask, PBS, DMSO, and other reagents. The first thing we wanted to think about is we need to warm up stuff at the first, including our media, and we need to go to get the biological safety cabinet ready. When this stuff warm up, I'm gonna go ahead and make the media. We also have the cells that we brought down in nitrogen liquid tank in our -80-degree freezers that will also thaw and bring in on plates. It is also better if you make the complete media prior to use. Do not freeze thaw serum every time. Make 10 ml aliquot of it and store some at 4 degrees and keep some frozen for later use. This will minimize the risk of contamination and as well as protein denaturations. For different stage of adherence, we prepare the desired medium. The initial stage of the adherence conducted with absence of FES and use of low glucose medium. This medium known as the incomplete low glucose DMEM medium. When this media prepared, we're going to get the cells from the -80-degree freezers. And I'm gonna go ahead, stick them into water bath. While they thaw, I go ahead and I do the rest of the protocol. And we also go ahead and add some DMEM complete media to the flask. And then I shoot the cells into the medium and mix them. And we get the cells put into the tube for centrifuge. After the centrifuge, you can see it's a small plate at the bottom of the tube. We get out the media at the top of these cells and resuspend the cells into the complete DMEM medium and add to the flask and incubated this flask into incubator at 37 degrees Celsius and 5% of CO2. After 24 hours, we assess the growth and viability of cells, and after 72 hours, we get out the media, wash the cells. We wash because some protein in serum will inactivate the trypsin, and then we add trypsin to the cells and incubate it for some minutes. After some minutes, when the cells detach from the flask, we collected the cell suspension into the tube for centrifuge. After centrifuge, the first thing we wanted to do is we cell count by trypan blue staining for plates. And then after that, we add 1 ml of DMEM complete medium, include the number of desired cells into each well of 12 well plates. And then we incubated this plate in incubator at 37 degrees Celsius and 5% of CO2. After 24 hours, we get out the media, wash each well, and we have already prepared the overnight culture of vibrio cholera in LB medium that we centrifuge these cultures and then wash with PBS and resuspend the pellet of vibrio cholera into incomplete low glucose medium without antibiotics. And then we add 1 ml of these suspensions into each well of 12 well plates that contain cells with 17 to 80 confluency. And we incubated this well for 45 minutes in incubator at 37 degrees Celsius and 5% of CO2. After 45 minutes, we get out the medium of these infected cells and wash with PBS for removing the unadherent bacteria. We're washing 4 times with PBS, and then we add 1 ml of Triton X100 to each well and incubated 2 minutes at room temperature. After 2 minutes, when the cell lines lysed, we collected the cell suspension into the microtube and centrifuge this tube. After centrifuge, we get out the media and then resuspend the pellet into the PBS. And in the final step, the serial dilution was plated for determining the number of adherents of bacteria. The result of colony counts presented in the table for calculating the percentage of adherence percent, we must divide the number of CFU of adherent bacteria by the number of CFU of the inoculum. In our study, in MOI35, we have the 57% of adherent. In MOI of 55, we have the 25% of adherence. And in MOI of 126, we have the 17.6% of adherence. Decreased amount of adherence in highest MOI shows that adherence in vibrio cholera depends on the concentration dependence manner. Thank you for listening to my presentation. Thank you so much.